Didn't see you there. Hi, I'm Dr. Leutrim, and I'm here today to talk to you about the significance of a Winogrossi column. Now, I'm 100% certain you've never heard of a Winogrossi column, and if you say you do, I think you're lying. Now, for those of you who aren't lying, you may be wondering what a Winogrossi column is. Well, I'm glad you asked. Pioneered in the late 1890s by Sergius Winogrossi and Martinez Berenic, the column is used to culture the organisms usually found in mud. Most of them, if not all of them, are prokaryotes. Now, you start with your Winogrossi column, which, as an example, is like this cut of 7-Up bottle. It will be longer, slightly more slender, and will have a harder outside. You will take your mud, preferably from a pond or puddle, and add it almost 3/4 of the way up of your Winogrossi column and compact it, and then add your carbon and sulfur sources, calcium carbonate, paper, and sulfur carbonate. If you need to add more water, if necessary, go ahead. Now, once you're done adding everything to your Winogrossi column, you'll enclose it using aluminum foil or a nice plastic wrap, and then put it by your window so it can be exposed to the sun. After months of allowing the bacteria to culture, you will see different colors begin to develop. Using this, you'll be able to determine what kind of different cellular respiration or fermentation the bacteria went under, and you can tell by what their byproducts are.
Welcome back, everyone, to tonight's Who Wants to Be a Millionaire? Today's show is a microbiology special, and our contestant, Arnold, will be answering questions on the Winogrossi column. Arnold, are you ready?
Sure. What are my lifelines?
Your lifelines are 50-50 and call an expert. You can get the 50-50 will do, but a call an expert will let you hear about an expert.
Let's do this.
Okay, I'll start off with a lightning round.
Wait, what?
I'm going to ask you the question, and you'll answer as quickly as possible. For the sake of time, I won't give you any answer choices to help you.
What is this?
First question. What is the purpose of the Winogrossi column?
Oh, it's to understand how microorganisms interact and develop their environment.
Correct. How does the column demonstrate this purpose?
Through microbial succession.
What is microbial succession?
The process by which microbes alter their environment, making it more suitable for other microbes to inhabit.
Just like a textbook. Next question. How do you make a Winogrossi column?
Well, you put the shredded newspaper first, then fresh soil and pond water so that the column is full more than halfway. The column should be sealed with plastic to prevent moisture from escaping the column.
Okay, the lightning round is over. Now for some tougher questions. As the cellulose promotes rapid microbial growth, what is depleted in the sediment and why? A. Carbon dioxide, B. Oxygen, C. Sulfur, or D. Nitrogen?
Oxygen.
Which area remains aerated? The bottom, lower half, upper half, or top?
The top of the column, because oxygen diffuses slowly through water.
What type of processes permit microbes to live in the bottom of the column? A. Fermentation, anaerobic respiration. B. Photosynthesis, anaerobic respiration. C. Fermentation and anaerobic respiration. Or D. Chemosynthesis and anaerobic respiration.
Well, since there's no oxygen at the bottom, only microbes that use fermentation or anaerobic respiration can live at the bottom. So stirring exists here as well because oxygen harms it, so I have to go with C.
You will be correct. Moving on. The bacterium desulfur vibrio produces what type of gas when utilizing the fermentation product for anaerobic respiration? A. H2S, B. CO2, C. SO2, or D. H2O. And for those at home, that's hydrogen sulfide, carbon dioxide, sulfide dioxide, and come on, water.
It produces H2S.
What process does the H2S enable for other bacteria? A. Chemosynthesis, B. Photosynthesis, C. Aerobic respiration, or D. Anaerobic respiration.
The gas enables sulfur-producing bacteria to carry out anaerobic photosynthesis. These bacteria produce sulfur instead of oxygen.
What color bands appear in this region of bacteria? A. Green, B. Purple, C. Green and purple, or D. None of the mentioned.
A green band and a purple band. So I'll go with C. Both are above the sediment at the bottom.
Wow, specific and correct. Well then, the bright red region of the column above the sulfur bacteria holds what type of microbes? A. Sulfur-reducing bacteria, B. Cyanobacteria, C. Clostridium, or D. Non-sulfur-reducing bacteria.
It holds purple non-sulfur bacteria, and they are photoheterotrophs because they use organic acids as carbon source. They cannot live in areas of concentrated H2S, explaining why they appear above the sulfur-reducing bacteria.
Trying to answer the question before I give that? Well, let's see, try this one. Which type of bacteria located in the aerobic zone of the column can process any H2S that diffuses to this region? A. Chemoheterotrophs, B. Photoautotrophs, C. Chemoautotrophs, or D. Photoheterotrophs.
Well, I know sulfur oxidizing bacteria are in this region, but they don't produce H2S. If they produce CO2, then the answer is chemoautotrophs.
Correct. At the top of the column are aerobic bacteria responsible for oxygenating the water.
I want to use 50/50.
What?
I want to use my lifeline.
Fine. Between A. Desulfovibrio, and C. Cyanobacteria, which is found near the top of the column?
Cyanobacteria.
Nice guess. What three factors determine which area microbes occupy in the column? A. Carbon source, B. Energy requirements, C. Environment, and D. Oops. Looks like we're out of time, folks. Tune in next week for a completely different show.
No!